I would like to welcome you all. I will be talking about clinical implications of stem cell as therapeutic targeting in schizophrenia. I will be talking about the stem cell technique firstly, and then I will be talking about the project GWASS, and because it's very interrelated. And I will be talking about schizophrenia and stem cell applications with three subjects, and then I will be talking about drug discovery, and then I will finish my presentation. When we have a look at the stem cell technique, as our professors mentioned, well, a biopsy is taken from a patient, and the patient's cells are reprogrammed into pluripotent stem cells, and after a conversion to another cell, then with monitoring in terms of disease, we can use it for understanding the disease mechanism, and for the improvement during therapy, it might be a good application. I would like to talk about the genome project called GWAS, and the USA was a pioneer, and it's used in the research of single nucleotide polymorphisms, and according to this, in what genes we see the defects more. This was the main question. And then in schizophrenia, we have the screenings of up to 40%, 40,000% of the patient. We get the CMPs. And for the CMPs are important for the development of neurons, and these CMPs have various effects. But which CMP is actually included in the disease mechanism? We do not have exact information, so we have a omnigenic hypothesis, and it says that gene regulatory networks are sufficiently interconnected, and that all genes expressed in disease-relevant cells contribute to the functions of core diseases. So, why do we use stem cells? Well, the main aim of stem cells actually is related to the development of the diagnosis of schizophrenia. I mean the localization of the pathogenesis is very important, so CMP variants can be monitored, and we can see the level of the mutation, degree of the mutation. It's very significant. So let's have a short outlook historically. Stem cell, after the beginning of 2000s, was started to be studied, and after 2010 it's changed its dimension, and then we have more rapid clinical studies, and 2011 it was included in the area of psychiatry, and with schizophrenia it was thanks to the schizophrenia, it was in the Department of Psychology, because it's a very complex disease. And then we had the recovery of glutanergic systems, and then about serotonergic system, we have some recent studies, and we see very good publications related to the depression. So, what is the level of stem cell applications today? Until 2019, we have sources of literature up to 280. And we have some indirect studies. It might be animal studies or human studies. We have a wide pathogenesis focus on schizophrenia, so I have three main topics. One is the relationship between differentiation, the other one is microRNAs, and the last one is the oxidative stress findings. What all of these indicate, we will be looking at this because all of the studies are on the microRNA, but on glial cells we do not have sufficient data. I mean, I see a recent study of oligodendrocytes, but I think the data is not sufficient. Well, in terms of neurodevelopment and differentiation, the first initial studies show that Shang and colleagues showed that in 3 patients that this one cell is monitored in a stem cell level, and this is a verification study that repeats itself. It's not that effective, but I think it's important. But after that, this study is the milestone in this field. And actually, 3 patients and 3 control group patients are included in the system. We have a PCB95 protein. In this protein group, there is a defect, and it causes a glutamate receptor expression. And in addition to that, the intracellular communication is also defected. And before the stem cell studies, and the signaling pathway actually is defected, especially in bipolar disorder. It's important because after the treatment in this step, loxapine treatment can be an option. When it's used after the treatment of loxapine, WMT signaling pathway is corrected. So the problem is corrected with the loxapine, and I think it's very significant in terms of treatment. It's a very promising step, and in 2011 it started to be used. But we are using antipsychotics for treatment, and including clozapine, and no other similar antipsychotic has this converting effect. The others were unsuccessful, and when we see the dopaminergic system, we have some defects, and I think we know that they have a key role in the development of schizophrenia. And according to the main mechanisms in the dopaminergic neurons, we actually see the developmental stage of schizophrenia. Well, the dopaminergic neurons, not just in the midbrain, but the changes in the neuron in other areas of the brain are significant for us. The studies show that in the dopaminergic system we have controversial hypotheses. We have lots of question marks because there's an increase in the dopamine level, but especially in the midbrain, especially in the mesolimbic area, there is no correlation. I mean, there is no correlated neuron increase, and on the contrary, there is a decrease in the number of neurons. So, in addition to that, when we focus on that subject specially, we have a FOXA2 protein, and well, we can control the dopaminergic midbrain neurons. We can monitor these neurons, and after the monitoring process on the midbrain, similar to the other studies, there is a decrease on the dopaminergic neurons or no significant difference at all. So, dopaminergic system is a system that we actually lead our studies according to that system, and I think future studies are needed to develop refined differentiation protocols. And we need to resolve the issue of altered dopaminergic function across various brain regions, because we don't know the source of it. I mean, it might be the midbrain or other regions in the brain. We don't have sufficient data about it. So what about glutamatergic neurons, especially in the last 10 years? The pathogenesis is more effective on the last 10 years in schizophrenia. So the basic mechanism affects the glutamatergic neuron system. So we say that we have sufficient level of studies, and there is no surprising data, especially in according to a dysfunction. We do not we have a significant difference between the control groups. So, as I mentioned, in the norepinephrine and epinephrine is significantly increased, and we see it on the imaging modalities too. There is an increase. So this is a significant correlation between them, but the increase might not be compliant, and this may be another research topic for us. Structurally, the hippocampus area, especially in terms of schizophrenia, it's one of the most discussed topics, and I think it's the one that we blame for the most. And what do the stem cell studies show? Well, it's not a different hypothesis, actually. It says that after the stem cell application, if we see an early delay of differentiation between the prefrontal cortex and hippocampus, the synaptic connections are delayed. So we have an impaired network connectivity. And the interesting thing is, while monitoring the stem cell applications and in the sodium-potassium channels, we have a mechanismal pathology. So in the gyrus, we have a cognitive disorder related deficiencies, and in this kind of patient groups, in the memory areas, we have some impairments too. This is the suggestion. So let's talk about the animal studies. The main aim is that, well, normally each cell is a stem cell in the embryonic term, and after this term, actually after 2010, in adults we have stem cells, and stem cells are localized in the hippocampus, and it actually has a role in regenerations, the studies suggested us. And as you can see, this differentiation can happen in adult period, not just in the embryonic period. And a disease has an epigenetic aspect too. So in the adult period, the epigenetic defects can impair the stem cells, so they can be effective in the pathology of schizophrenia, especially in the adult neurogenesis. This gene is one of the most blamed genes. Well, and after the genome project, mRNAs are also an interesting topic in this patient group, especially in terms of the maturation of the genomes. And in the adult brain, mRNAs are also a risky group in ASD, for example. In a study, they found that MIR 137 gain of function causes downregulation of the presynaptic target genes, and it has a role in cognitive impairment. So I would like to mention this part because the alterations in mRNA, how can they be corrected? We have a CAS9 method, and with the correction of this impairment after remonitoring, the hippocampal memorial impairments are corrected. The CRISPR is one of the most important topics today, because it makes the repair possible thanks to that. And I think these studies, especially on the SNPs, show that with this application, the mutations on the hippocampus, the dendrites are corrected. So finally, mRNA networks enriched and SCZ risk mRNAs may be disrupted at a global scale. It's a genetic disease, so maybe we should focus on it more. The third one is the oxidative stress, because when we look at the oxidative stress, it's not a specific finding, because in each disease we have an energy impairment. And in the MI spectroscopy studies, the psychotic diseases and affective diseases, we see it very commonly. But when we check the studies, oxidative stress, it will be more significant with the stem cell applications, because after the differentiation of the cell and to the last stage, the impairment on oxidative stress is seen, and the formation is disrupted, as the studies suggest us. And the main point is with the microblasts, the oxygen species and radicals are increased, and according to this, the cellular destruction is more, and this is the area that is more affected. And I would like to underline that it's one of the areas that we use the medications more. And in the studies, after usage of the valproic acid in the oxygen radicals, we have a significant decrease, and the distracted cells have more increased rate of degeneration, and I think it's very important in terms of treatment. Another point in oxidative stress is that the quantity of membrane level, there's a thinning, and the decrease of the membrane potential leads to an impairment of energy metabolism in schizophrenia patients, and it actually causes more necrotic phases. And there's another hypothesis. Can we have a transfer between mitochondria, and maybe it can be transferred to a impaired cell, and after the transfer, instead of this impaired areas, the healthy mitochondria is seen. and I think it's one of the most significant modalities. In a person, if we see a pathogenesis related to oxidative stress, with this application, the improvement may be seen, and I think it's a promising one. So what about the drug development? We have multiple pathways here. The post-mortem studies, imaging modalities, and the CSF examples. With all of these, the stem cell applications are also included, and we have a very wide range of drug exploration, and especially in schizophrenia. As I mentioned earlier, loxapine has a good rate of improvement in WNT and other signaling pathways in SCZ and iPSC neurons. But maybe in other pathologic pathways we need to see its effect more, but we see that valproate in the oxidative stress impairment, it has a very regulatory effects, and CRISPR and Cas9 in schizophrenia patient groups and in mRNA impairments and genetic mutations. The mutated area is extracted and the treatment is done. Maybe this might be a precursor for the treatment of schizophrenia. And here, as you can see, this is the summary of all schizophrenia stem cell application studies. Well, this is a case that can only be conducted in a very limited patient groups, like 4 patients or 3 patients. They have a very limited cohort. Well, maybe if we can increase the number of the patients, we can have sufficient data. In the they say we want to work on a specific cellular area and we cannot predict the impairment earlier, so maybe the studies repeat themselves, especially if we want to work on brain glutaminergic region. We have some factors. Maybe we can look for the specific impairment in that person related to the glutamatergic regions, but it's costly and it's very challenging to focus on just one specific area, the region of the brain. And there are some alteration findings. Well, to conclude, what has been found up to now, we have a decreased migration, and there's an increased non-canonical WNT signaling, and we have an elevation in oxidative stress, and according to that we have the mitochondrial damage on the forefront, and we have some differentiations. To conclude, well, the transformative discovery of IPSS has widely opened the door for new translational strategies. We cannot get an animal or human brain directly. We cannot see the brain of the schizophrenic patient, but in suspicion points we can see the level of impairment or the level of pathology in that patient, and the individualized approach of monitoring is important, and I think we are at the beginning of the process. We need it. That's what I would like to underline, and I would like to invite you to our Congress in 2020. Thank you for your patience.